had a basic discussion about that, what autocrine signaling is all about, what paracrine is, endocrine and juxtacrine. So today we'll be discussing them in detail, and then we'll move forward. So talking about the autocrine signaling. So autocrine, auto means self. So when one cell have the effect over itself, that is what autocrine signaling is all about. So the signaling molecules produce the effect on the same cell that produces it. That means we have this cell produces some signals, and these signals work upon the same cells. It forms it is a form in which the cell secretes a hormone or an autocrine agent that binds to the autocrine receptor on the same cell, leading to the changes in the cell. So you can say we have this particular cell and we have the receptor for this particular, for the ligand which have to come and bind. So the ligand comes from within the cell itself. It comes and bind over this receptor, and there occurs the complete cell signaling process. Examples of these are autokines, cytokines, immune cells, and the growth factors. Now the second one is paracrine signaling. So paracrine, remember it as parallel. So parallel means jo uske nearby. It is located in the nearby area. So the action occurs in the nearby cell. The cell-to-cell communication produces signals to induce changes in the nearby cell. Close proximity, that means we have this cell, and in a close proximity of this cell, we have another cell on which the ligand would come and bind. So this one is autocrine. In autocrine, we have the sensing or the signaling over the cell itself, the same cell. In paracrine, towards or in the close proximity, the functioning would occur. They are secreted in the immediate environment. Example for these are fibroblast growth factors, hedgehog family, VNT family. Hedgehog and VNT are both in the Drosophila. When we would be studying about Drosophila, in that particular thing, we'll be discussing this particular topic. Then we have TGF beta family, neurotransmitter action between the nerve cells and the synapses. So if you all remember, let's consider this is one nerve cell, this is another nerve cell, and the junction between the two nerve cells is what is known as synapse. So when the first nerve cell releases its signal, in the gap between or the space between the starting point of the next neuron, this particular point, this particular type of cell signaling is what is the example of paracrine signaling. The third one we have is the endocrine signaling. So in the endocrine signaling, the endocrine system works. So if we have learned in class 11th or 12th, we have a system known as endocrine system, in which the blood is used as a stream so that the signal could be passed on to a far located cell. So the signaling molecule acts on the target cell distinctively located from the site of synthesis. The most common example is we have the growth factors. So they are mainly produced from the pituitary gland, and from that they move those signals of growth, travels from the brain area towards the area where it need to actually work upon. So that is the most common example you can remember about. So in this type, there occurs long range signaling, which is transported via the blood stream. This blood streaming concept is very important. Then the examples for the same are, first we have endocrine hormones like the insulin, which is produced from the pancreas. Then we have testosterone, progesterone, gonadotropin. All these are growth hormones which are produced from the pituitary gland. The signal for these growth hormones is produced from the pituitary gland. Now, the next point is about the juxtacrine signaling. So, juxtacrine signaling, what is the most important point you have to remember? That it is contact dependent. That means we have one cell, we have the second cell, and for the signaling process to get completed, for the signaling process to have the function over the complete scenario, there need to be physical contact, or it is contact dependent. So it requires physical contact with the target cells. The type of juxtacrine signaling molecules, there are various types of juxtacrine signaling molecules. The very first one is membrane ligand, membrane protein. So The example for this is delta notch pathway, which we would be studying in the subsequent slides. Then we have the second one, the communicating junction link between the intracellular compartments, example plasmodesmata. Question for you is, where is plasmodesmata present and what is the main function of plasmodesmata? Anyone?
Present in plant.
Okay, very good. What is the main function of plasmodesmata?
Water
Exchange of
transport.
material.
Yes, very good. If you all remember about जो एक signaling, we have a signaling like apoplast and symplast.
Yes, ma'am.
Yes, so
Symplast is through plasmodesmata.
yes, I was saying the same thing. In the case of symplast junction, in the case of symplast transport, plasmodesmata help. So the third type is the ECM, membrane protein, example cadherin. So these are the types of the ligand. These are the types of the juxtaprint signaling molecules. Now this was about it. Let's discuss about the types of ligands now. So types of ligand. Yesterday we discussed about what is a ligand and what is a receptor. Just anyone try to just define what is a ligand and what is a receptor. Then we will move forward to the types of them. Yesterday we have discussed about this.
Ligand simply means the signaling molecule and receptors in which that ligand will bind.
Okay, yesterday we compared this particular thing with which example?
Stylus and its stand.
Yes, very good. So yesterday we discussed about this particular stand and this stylus. So this particular stand of the stylus is actually the receptor, and the ligand, which is this particular stylus, it would come and bind to this particular receptor area so that the complete functioning could take place. So this was about the ligand and the receptor. You can you can connect it with this thing as well. You have a bowl and you have a ball. So that particular bowl is the receptor and this ball would act as a ligand. So if you want to draw it this way, you can draw it like this is what a receptor looks like. It is like a bowl and